Okay, so for today's lessons, we are going to look at this indirect active transport. Okay, so compared to the direct active transport, indirect active transport, you can see that the involvement of ATP is not directly. Okay, I mean the energy release for the ATP from the ATP, the hydrolysis of ATP does not involve directly in the transportations of the particle that we want to move them into the cells or out of the cells. Okay, so if you look at these diagrams, you realize that the transportation of proton, yes, we pump out the protons, but proton after that will diffuse back. Can you see that? Okay, by facilitated diffusion. So from these diagrams, you will see that ATP first phosphorylate the proton pump out the proton. So proton will move from the cytoplasm into the cell wall, okay, in this structure. So write down this part is a cell wall. This one is the cytoplasm. And we do know that in order to transport the sucrose in, we cannot allow the sucrose actually move into the cells by facilitated diffusion. Impossible, because right now sucrose is water soluble. Okay, they are water soluble and hydrophilic. So if the water soluble and hydrophilic, impossible for them, and they are large also, they cannot pass through. Okay, so for them to pass through or diffuse across the phospholipid bilayers is impossible. Okay, so how we transport them in? So very simple, we do have these kinds of proton pump. So ATP phosphorylate the proton pump. So we pump the protons out. So this is active transport. Because why? Can you see that? Low concentration of proton, high concentration of proton. So means that as long as I have ATP, I will be able to pump the proton out of the cells, right, against the concentration gradient. So if I label here the first one, so first one actually is the active transport of proton, so against the concentration gradient. Okay, so there is an accumulations of the protons in the cell wall compared to the cells. So protons actually want to move back because higher concentration, lower concentration, and a lot of positively charged ions here. Here, lack of the positively charged ions. So therefore, they want to move back. So if one to move back, the main condition, you move back, you bring in the sucrose. So means that proton have to bind to the binding sites on the sucrose proton co-transporter, and then sucrose bind, both of them will move into the cells together. And do you realize that in this part, okay, if I label here process number 2 and process number 3, 2 and 3 they do not involve, both of them do not involve the phosphorylations of the ATP. So therefore, number 2 and number 3, both of them are facilitated diffusion. Okay, because involve the transport proteins. Co-transporter is a transport protein. But for proton, number 2, proton back because high concentration, low concentration. So therefore, for proton move down their concentration gradients. Okay, but how about sucrose? Sucrose actually independent of the concentration gradients. Why? Because for sucrose to move in, they need a proton. So they depends on the proton. So as long as we do have the proton at the cell wall, when proton move into the cells, then sucrose molecule will move in as well. So it means that we can actually have the sucrose concentration, even though higher inside the cell, the sucrose still can move in because it depends on the availability of the protons. So therefore, in this case, we can achieve something like against the concentration gradients. Are you clear? Okay, so this is something special where facet diffusion against the concentration gradient, which is special because in this case, sucrose move in actually dependence of the proton. But overall process, what is the aim of the overall process? The overall process, we want to move the sucrose into the cells, right? Overall process, can you see that? If one out, one in, it considers 0. Move one out, move one in. I give you 10 ringgit, I take back 10 ringgit. So means that you still get nothing. Correct or not? I also didn't lose anything. Correct or not? But in this process, sucrose move in. Can you see that? Are you clear? Sucrose move in. So the overall process is to bring the sucrose in, and the overall process also require ATP. So therefore, the overall process is termed as active transport. But in this case, it's an indirect active transport. Are you clear? Okay, so let's look at how we write it. So okay, this is how we write it. For proton pump number 1, so ATP phosphorylates the proton pump. Therefore, the protons are pumped from the cytoplasm into the cell wall against its concentration gradients. Okay. So number 2 involve the co-transporter. So first, proton have to bind to its binding site on the co-transporter. Sucrose also bind to its binding sites on the co-transporter. But H+ move into the cells by facet diffusion down its concentration gradient. On the other hand, sucrose move into the cells by facet diffusion, but again its concentration gradients. Okay, so this is a slight difference when we look at facet diffusion that can against its concentration gradient, because sucrose move into the cells actually depends on the availability of the protons. Okay,